Hello everyone, my name is Anfam and I am going to present to you about the reverse transcript polymerase chain reaction. Reverse transcript polymerase chain reaction is commonly used to detect the presence of mRNA, pre-mRNA, or any other type of RNA. In some cases, polymerase chain reverse transcript polymerase chain reaction can be used to detect a virus with an RNA genome, such as the HIV virus. So first, let's talk about the principle of this technique. Standard polymerase chain reaction is a fast and inexpensive technique that make copy of specific DNA segment. They are also known as the molecular photocopying. So with this technique, many copy of DNA can be replicated in just a few hours compared to the traditional method of that the cell used to replicate DNA template would take very long time. RT PCR technique is basically the same with standard PCR, but was modified in which they use mRNA as the starting template instead of DNA. In general, we have learned about the transcription and translation in gene expression process that the DNA was transcribed into mRNA and then translate into protein. But reverse transcription switch out the first step, which the the reverse transcriptase enzyme use mRNA as the template to produce complementary single-strand cDNA. From here, cDNA can be used as the template for PCR reaction. The reason why we perform reverse transcription PCR instead of normal PCR is that some cell has DNA but they're not actually produced by the cell. Also, it is very specific when using specific parameter in transcription process of cDNA. Therefore, RT-PCR is a good outer way to confirm the presence of genetic material. There are some good advantages of RT-PCR such as they have a high sensitivity due to exponential amplification of the RNA template, and they also make DNA RNA extraction more efficient. As I said, RT-PCR typically used to amplify and detect the RNA gene. Usually, RT-PCR can use in retrovirus that the virus use reverse transcriptase enzyme to convert their RNA into DNA genome. This figure shows an example of retrovirus, which is HIV virus. When the HIV cell fused with the white blood cell, which play an important role in the immune system, the virus the virus will then release its RNA into the cell. The reverse transcriptase enzyme will bind with the RNA from the virus and convert into DNA. From there, HIV DNA will then replicate and reproduce during the cell division. And in a research that RT-PCR was involved to detect HIV RNA in HIV positive patients in the Republic of Macedonia. According to this research, 33 patients were selected and divided into two groups. 13 people were HIV positive and 20 people were healthy blood donor. RNA from the whole blood cell was extracted and using RT-PCR kit. The results shown that none of the healthy blood donors have HIV RNA present. And this figure one here showing the electrophoresis result of the amplified sample. The positive HIV band at 210 base pair for all positive control and negative and positive sample. In figure 2, the result of HIV RNA was detected in 9 out of 13 patients. Um, the other patient which is negative is patient number 7, patient number 9, 11, and 12. They, according to the discussion of the research, they explained that one of the patient is currently intravenous drug user, and 2 other patients are homosexual gene. From these data, we can conclude that RT PCR is a very reliable technique for the detection of HIV RNA. It is also inexpensive and can be easily conducted in the lab. Okay, my question related to my topic is HIV virus take advantage of reverse transcriptase enzyme to transcript their RNA into DNA for replication. Is there any way that we can inhibit the enzyme so HIV is not infectable? This is my reference. Thank you for listening.